So what I'm sort of talking, so I'm from Roche Pharmaceuticals. I was really sort of wanting to talk about the big data and the type of data we're just starting to see come into our industry and the sort of impact that may be having in the future and the opportunities that may be presenting to us. I think maybe unlike sort of our previous speaker, who sort of presented a lot of nicely solutions to problems, I'm really just been presenting the problems and the opportunities. On the plus side, I'm going to say we've got no theorems and I'm going to stick to that promise. But it's sort of in a real sort of situation, which is sort of my picture on the right-hand side, that actually I see something that we're in a situation, it's been a long, hard walk to get here, but looking forwards, what I'm sort of seeing is an explosion of various different sorts of data coming forwards, and sort of just starting to think, I think, with the industry as a whole, about how we deal with these and how we work with these in the future. So just sort of an overview of what I was going to talk about. I'm just going to give a little bit of context and background about the pharmaceutical industry, because I realise it's probably, a lot of people in the room won't have any much knowledge of that at all. And talk about sort of some of the existing challenges and really sort of things which are driving some of the changes we're seeing going forwards. And then sort of talk a little bit about our initial responses and some of the first steps and things which have been going forwards, and then maybe how that is going to develop and sort of explode and grow further in the future in different new challenges and new opportunities. So just a sort of a background with the pharmaceutical industry, and this is sort of just giving an example of Roche as just sort of an example company. And it's really sort of to make the emphasis that this is really a very big, very global enterprise that we're sort of dealing with here. So we've got figures here. So we sort of talk about that, 137 million people being treated with some of our drugs or medicines. We're talking over the course of the year, 19 billion different tests being carried out. Again, so huge numbers of people involved in sort of that type of dimension. The pharmaceutical industry is also one of the heaviest sort of industries in terms of investments in R&D worldwide. So we sort of see, if we talk about 90,000 employees and sort of, getting on for a quarter of those employed in R&D efforts, and sort of spending a total of about $10 billion in R&D during the course of the year. So sort of a high teens investment and turnover in terms of R&D. Again, very high compared to most industries. And the majority of our sort of spend in R&D actually sort of goes within running clinical trials. So really the experiments of where drugs are working, how working, what the effects of drugs are on patients and people. So we're sort of talking, in 1 year we're sort of running in the order of 1,500 different trials and involving around 300,000 different patients in there. You can sort of see the sort of scale of data the work we're doing with here is quite large. It's sort of covering, again, a number of different diseases, and these diseases all sort of can be, have similarities of course, but they also are quite different from each other. Roche is kind of like probably the leading company in terms of oncology and haematology, and in terms of diseases, they've probably been one of the ones which have sort of been driving some of the changes that I'll talk about later in sort of thinking about personalised therapies the most, in that they're ones which are very defined diseases. They're sort of quite clear-cut and quite what they are. We're also in sort of areas like infectious diseases, which have sort of infection, can be very targeted as well, and that different types of infections have different types of treatments. But also sort of in some of the areas which their diseases tend to be less well defined. So we talk about immunology, which is mentioned by our first speaker this morning. So there's a whole range of autoimmune diseases, things like lupus, irritable bowel disease, rheumatoid arthritis. And actually the reasons for these can be very much harder to determine. So the treatment there's a whole range of different treatments which can be applied. And actually sort of what the drivers and everything that goes on seems to be a very multi-dimensional, probably a huge number of different factors in those diseases. Similarly, the area of neuroscience, I think this is something we recognise going into sort of the future with an ageing population. Conditions like Alzheimer's, conditions like Parkinson's disease are going to be increasingly important. But actually our understanding of those diseases is sort of lacking a long way behind some of the sort of major, more studied, more conventional diseases. A lot more work's required there. What's also interesting about Roche as a pharmaceutical, as a company, is that as well as producing the pharmaceutical, so as actually the medicines, it also produces another arm which is actually running the diagnostic tests. And these are sort of, are clearly another key part in sort of the healthcare pathway you should go through. And these cover a whole range of different activities. These go from sort of very simple lab blood tests you may get if you just go to the doctors, the outpatients, the hospital, very regular treatments. They consider things like diabetes patients who are constantly having to monitor their levels and performing a large number of tests. But it's also moving to areas which are sort of like the more advanced, more newer techniques, which are sort of looking at molecular diagnostics. So again, in terms of sort of the first speaker this morning, it's sort of measuring things like measuring the gene expression levels, the proteome levels, look at the gene sequencing. So again, much more modern technologies within there. But again, this is potentially things which are generating a huge amount of information and a huge amount of knowledge about patients as we're providing all of these. And one of sort of the concepts which have come from this is sort of the idea around personalised healthcare. And this is really sort of the idea, instead of having a one-size-fits-all solution for a treatment of a particular disease, that for any patient who's got a particular disease, they would receive that treatment. So this can be tailored a lot more to any individual's patient's needs, both in terms of the actual treatment they get, but maybe sort of in things like the dosing level that they get, the timing and the scheduling of the dosing that they get. And the idea being that actually the two different arms we talk of the industry here, is both sort of the treatment medicines and the diagnostics, can interact very closely together in here. That we're able to select the right patients based on the diagnostics, give those patients the treatment, and sort of generate that information. And that can sort of, and the information we generate from those, and we can measure in diagnostics and help to feed to refine that information going forwards. I guess just sort of now just moving a little bit onto the traditional way of doing drug development. What we sort of talk about is having a drug development pipeline. And sort of the idea of this is it really starts over on the far end there, other side there, with sort of the basic research, and then doing drug discovery. So this can be actually sort of the traditional way of doing this was through high throughput screening, where effectively millions of different potential molecules were tested in a sort of in vitro type of situation. There's different modalities and different approaches to this now as well. It would then sort of go through one of those once you found something potentially of interest in the laboratory, some preclinical type of testing sort of just ensure the safety within sort of an in vivo environment. And then we sort of went through a series of clinical trials, so the imaginatively phase 1, phase 2, phase 3 of clinical trials. As we're doing this, we're sort of increasing the number of people who are actually enrolled in these trials, number of patients they're being tested upon. And it's probably sort of a rough guideline that sort of each of these different vertical bars here would sort of be a time period of around 2 to 3 years. So you can sort of see by the time we go from the left to the right hand side of the graph here, we're actually sort of talking a long period of time in the development of drugs. After all that has been done and hopefully we've got positive results, they're then submitted for a regulatory authority who has to sort of actually independently review these to make sure that they're approved, can be used within the patients. There's also another step in which we have to sort of get reimbursements. This is by organisations such as NICE or insurance companies within America who actually sort of say, is it sort of going to be cost effective to be able to use as well as just being efficacious. So it's obviously required for approval by them before it can be actually used. And then it could sort of actually be taken out into, given to patients, hopefully treating patients. But then there's also typically always been this case that there would be post-approval trials going as well. So continuing to study the drug going forwards into the future when it's actually in clinical use, gaining more knowledge about the drug or just confirming some aspects of it. Now, one sort of aspect of these things is that one, I've sort of mentioned that these things are a long time to develop, a long time to develop a new medicine. Another aspect of this is, although it's called very much of a drug development pipeline, if you look at sort of the shape of those white lines at the top there, it really is more of a funnel. That actually, although we have a huge number of potential drugs which enter sort of human development, a lot of those drop out in the meantime. So actually only a small proportion of those actually reach the end and actually can be actually given to patients. So over on this side, we can sort of see we have the sort of the attrition rates, typical attrition rates, which we see at each individual stage of the development process. And here, this is sort of quoting that only sort of just under 10% of drugs actually make it from the starting end to the human to actually be able to sort of actually be marketed at the end of the day as approved drugs. The other aspect that's going on is sort of just the increased level of cost associated with developing a drug. So this is sort of a line which is just showing a trend from the 1950s up to the present day. And this is sort of the number of drugs which can be developed on the vertical axis, the number of drugs which can be developed for $1 billion R&D spent. And what we see is that on a log scale, that's sort of been effectively exponentially increasing, well, increasing cost, decreasing productivity over that long period of time. And now we're sort of talking in an era where it's sort of talking, and the estimates vary, but in the order of about $2 billion in order to develop a drug. Clearly, huge investment in order to take that forwards. So, okay. So we're clearly in the world where it sort of is a huge number of challenges going on in developing drugs. We're sort of looking in areas where we've got increasing competition, and one of the aspects here we're talking about really getting first to market has got a huge potential impact, because basically once a drug has been approved in a certain disease, it's going to get traction, it's going to get a lot of use, and it's going to be adopted. It's then very hard for similar drugs, even if they're slightly better, in order to get that sort of level of traction compared to drugs which physicians are already comfortable in dealing with. We've sort of seen the additional costs of doing things, but at the same time there's pressure that societies cannot continue to have the level of expenditure on drugs it sees at the moment. And underlying this is what we sort of always call the better than the Beatles principle. That actually what we're always in competition with is not just the best drug which has been developed at the moment, it's the best drug which has been developed for any disease since the start of development, which occurs all the time. This is equivalent sort of in the pop music world that, it's not just the pop music is not going to be at the top of the charts, the best song at the moment. It's going to be better than every past song which has ever been written previously, or else everyone just continuing to listen to the Beatles all the time. But understanding that as well, we've sort of in a situation where we've got the complexity of the biology that we're dealing with here. And this is sort of, again, something we're learning much more about, and it's getting a lot more complexity, which we're able to pose a challenge to work with, but actually gives us the opportunities as well. So some of the things which are sort of starting to occur, which are sort of as a response to these type of things. One is there's certainly sort of increased pressure about increased speed. So, for example, within the regulatory authorities, we've started to get ideas about accelerated approvals. And effectively what that means is when we're seeing sort of some very strong results from some of the very early trials, that they will actually give a conditional approval for that drug and make it much more widely available. At the same time, in parallel, we're running some of the later trials. So that provides effectively a quicker route to market, a quicker route to patients, some really sort of novel and innovative drugs. We're also sort of developing a number of ways of actually sort of be able to accelerate and make this development program faster or more efficient. So sort of things like novel trial designs, bringing adaptivity into them, either stopping them early when they're not working, or really taking them forwards and accelerating them when they're rapidly doing there, or making decisions as we're going along through the development program. I'll talk quite a bit more about sort of the ideas around personal health care, sort of matching up the patients to the right treatments later on. But there's also other things going on as well. We sort of look at clinical development decision support tools. Again, this is a linked concept, but it's giving sort of physicians the tools to be able to make the correct decisions and the information. And this is linked to areas around sharing of the data. That actually, we may be in a situation now in the global with the networking we can achieve, that actually information from one treatment, patients treated in one place, can be very informative elsewhere. And other ways of sharing that information around and forming this information. And we're also talking about different types of endpoints, so different things that we're measuring within the clinical trials. And particularly actually now getting more of a focus on not so much actually the treatment of the disease, but actually what it means to the patient in terms of that. And I'll give an example of that at the end. Underlying this has sort of been one of the things that we're talking about seeing is sort of developing technologies. And sort of I think the best example, really the poster example of this is really about genomic sequencing. So sort of understanding the human genome. Now it's only sort of about the beginning of this century that the human genome was first sequenced, and that was a huge international collaborative effort costing, quoted there $100 million. It took many years in order to do that. Many laboratories all around the world contributing to that. And what we've seen from that, and this is sort of plotted the cost per genome, is that this has fallen dramatically over time. At the same time, it's also become much more rapidly quicker and much more speedy in order to perform these analyses. And what we see, even compared to Moore's law, sort of the normal exponential improvement in computing speed, over the past decade, about a decade ago, it really was sort of hit a super exponential phase where the cost and availability of these technologies was accelerating beyond proportions. Interestingly, over the past couple of years, there's always been this boundary about doing the human genome sequencing for $1,000, and that was always one of the targets. Curiously, we seem to sort of just stop just above that, but I'm sure we'll reach there very soon. But as a result of this and the actual availability and the reducing cost and the more availability of these, this has sort of led to much more then widespread genomic collections, genomic sequencing collections going on. So sort of one of the high-profile ones within this country is one run by Genomics England, which is called the 100,000 Genome Project, which is exactly what it says on its label. It's really sort of trying to do 100,000, collect the whole genome sequence across 100,000 different individuals, which you imagine is a phenomenal amount of data in there. And currently, I just saw the other day, they've actually just passed halfway, so they've just reached 50,000 different genomes. We also see that there's various different commercial companies getting involved doing that as well. So there's one which is HLI, which is strongly involved with different commercial organisations, for example, sort of running genome sequencing, collaboration with clinical trials that's going on. There's another one, 23andMe, which was actually set up by the wife of one of the founders of Google. And effectively, this sort of provides one of these tests which you can go along to Superdrug and buy a little genomics test, and they come back and give you a few interesting different markers about whether you can curl your tongue or what colour your eyes are, things which you probably already know about yourself. And already has connections also to genealogy, so potentially you could find some sort of genealogy and understand where your roots come from. But also they're doing the thing where they're actually collecting all this information together and actually provides, now they've got a huge collection of genomic data from a huge number of individuals, and actually provides a great research tool that they can actually go and work with people in order to take things forward as well. So there's sort of very clearly this map going on that this is sort of driving the collection of very large data sets which can then be subsequently mined. And that sort of takes us sort of the area of personalised medicine, which is the idea that we can actually move from something where we've actually got a single treatment for a disease to ones which are much more moving from one size fits all to where it's actually sort of treatment decisions being fitted to individual patients' needs. And this sort of generally sort of has a few different things going on at the same time with it as well. One thing is that actually it changes our way of looking at diseases. So traditionally sort of diseases, particularly with sort of a cancer, one of the things people would be looking at is actually where in the body is the cancer as the main defining thing. So is it prostate, is it lung, is it breast, or whatever like that. The way it's moving now and the way actually treatments are starting to be approved and thought about is actually not so much there, but it's actually what's the molecular contribution of that cancer, so what's the diagnosis behind that. And actually recognise that the treatment of something which has got the same molecular driver behind it is going to be a much more common factor to cancer, whether it happens to be in the lung, in the kidney, or wherever, than actually the actual geographical part of the body which it's sitting in. And this is also linked to sort of the ideas around having a continuous learning and actually be able to use the data which we're collecting and all over the place in order to feedback and guide treatment and guide development of new drugs going forwards. And this is then actually sort of exchange the loop which we had before about having this link between the diagnostics and the medicines, but actually also bringing in data which we're getting from electronic health records, or the so-called real-world data, which is actually then sort of going to actually improve our information, give a much wider cohort in terms of the type of data that we're looking at, and actually sort of improve the database that we're seeing. And I guess sort of in terms of personalised healthcare, just to sort of sketch out the story, this generally started off with quite simple markers, so just sort of single markers that they were looking at. So the ones being here, for an example, if there was a certain gene mutation or a certain rearrangement of DNA, that indicated a certain specific sort of treatment, and we're able to develop particular treatments which would only work for people who had the cancer due to this particular driver, but would work very efficiently in this type of patient. And what that sort of meant is that, for example, this is for example for lung cancer, is that we were able to take the whole range of different lung cancers and segment that population to a whole range of different diseases, effectively, or sub-diseases, each of which would have their own individual treatment related to those, and actually very much of a focused treatment instead of just the general purpose chemotherapy. The thing about this diagram here is, you see over the blue side, the sort of blue half of it, these are ones which were currently unknown. So what we see is that, although we can explain the driver of a lot of these, there's also a lot of things which are very unknown within there. And I think what we're sort of experiencing and what we know from biology is probably that's going to be increasingly smaller and smaller subpopulations within there. I think we can get to those over time, but they'll just require much more study and even larger and larger populations to be able to see enough of these patients to identify them and develop any further treatments for them. As a result of that, that's sort of then driven the diagnostics market. So one of the things is, for example, Foundation Medicine, which is probably one of the leading companies for producing a general purpose wide-scale diagnostic for cancer treatments, is that they can now run a test which actually looks over all the different standard cancer genes, so all the genes that we know have some sort of drive or linked with the development of cancer. So I think it's in the order of 400 or 500 there. And from those, they're able to identify for any particular patient a list of the potential drivers which they see doing there. And then it also moves into being a bit of a clinical decision support tool. That for any of those, that represents certain different treatments which will be linked to those particular gene mutations. And even it can recommend potential clinical trials which are going on and very novel treatments which may be very related to those particular gene mutations, those particular molecular changes which you see in that particular patient. And also then this sort of then takes us from just having a simple single marker in order to identify the disease to having combinations of markers. So actually something where we're actually using some sort of measure, some sort of modelling algorithm to identify or to establish some sort of relationship within a panel of patients, a panel of genes, and outcome. So this is an example from a company called Genomic Health, which have a product called Oncotype DX, which actually sort of measures the probability that a patient who's been treated by chemotherapy would have a recurrence of their breast cancer over time, over a time period based upon different treatments that they have. And basically there's a score here which is actually some sort of formula based on around about the expression of 20 different genes, put through some sort of algorithm to give a score, and then they can read off what the risk of a progression or recurrence over a 5-year risk is. And then obviously that would then drive future treatments with things. People who are down this end, they'll just be continually monitored and viewed. The high-risk patients up at this end may consider some more intensive monitoring, some preemptive treatments, or maybe further surgery. And a similar sort of thing can be done. There's other sort of more of these type of methodologies coming forwards and approaches. But again, what's really interesting about this is that now these tests have been established and being run on an increasing number of patients, increasing number being used, and this is consistently growing and growing and growing on a daily basis. This is actually developing a huge database that we now have of people with their cancer patients with their genomic profiles. So the quote which is given here is over 180,000 of these genomic profiles, and I imagine that's out of date. So again, this is sort of something which is a great resource to be able to look at, to be examined in order to understand the disease, in order to be able to develop new treatments paradigms going forwards. Where it gets even more exciting still is that we can then start to integrate it with other databases. So for example, clinical databases here. And so the example I'll sort of just talk about for a little bit is one called Flatiron, which is sort of, and effectively what Flatiron is an electronic health record system. and which has been specifically designed to really sort of meet the needs of oncology treatment centres. And it's sort of, very well designed for that type of thing. But it's also been designed with the eye of actually be able to collate all that data from the trials from the centres being run, and actually to be able to use that actually as a data mining, provide that for information going forwards. So what we sort of see is that it's been used in the US, and it's sort of actually, widely used across a whole range of different cancer centres there. And effectively it's used those. It provides great benefit in terms of actually the cancer centres being able to efficiently run their sort of day-to-day routines, be able to function efficiently, but also collates together this very large database, so actually gives a huge amount of information, both sort of on the treatments, the way the treatments are being used by physicians, but also the outcomes, and the changes which people are making, and the different treatment pathways which people are following. So, what we're sort of seeing at the moment is this is sort of, over 2,500 oncologists with over 2,000 patients, but expanding on a much greater, on a regular basis. So growing all the time. And this is something that's only been around for a few years. And again, there's other similar sorts of things which are going on as well, some other similar sort of developments which are there. But what this potentially allows is actually now we've got the combination of actually this very wealth, the huge number of different patients we've got, so a very large N, with actually some very detailed information with a very large P in terms of sort of the tests we're running. And so actually sort of the interaction between those from things which are collected naturally during the course of a cancer treatment can actually enable us to do sort of some correlations between those and just identify relationships between the type of analyses with type of treatments you're being given, the genetic basis of those and the outcomes. There's obviously a lot of risk with these. These are not randomised trials or observational trials. We have to consider a lot of biases. There's a bias variability trade-off and potentially a lot of different analyses. There's certainly sort of a huge amount of information there. And then again, there's sort of other levels of complexity that actually what we're sort of talking with the cancer is that although we sort of talk about situations where we sort of think a cancer is a single thing, a homogeneous thing, in fact it's not. It's a very heterogeneous thing which is consistently evolving. And so what we see over time is that cancers will evolve, and they won't just evolve into one, grow, they won't just go into one thing, they'll have start evolving at the same time, have multiple different types of different cells within there, multiple different drivers with multiple different genetic expressions. And what this is showing as well is that actually with treatments, these can actually then very much change the molecular profile of cancers. So we may have a treatment, and the reason cancers evolves into multiple different types, different from where it started from. We have a treatment which actually kills off most of the cancer, but then there's this resistant bit, which is the yellow bit, which continues on and then profligates in the future. And that's what causes the drug resistance within oncology, which is one of the great problems. And so that sort of suggests the thing that rather instead of taking just a single measurement for any particular patient in order to get assessment of their molecular profile, do we have to be doing sort of multiple measures from this same patient? And this sort of gets the idea of doing single cell sequencing. Well, rather at the moment where we take a slice, so we take a whole plate of things and mush it all together. If we're actually able to work at the level of individual cells and take samples of individual cells, that gives us a whole profile within a patient of their molecular profiles, and then we can start to think about how that may develop over time and actually build in the treatment accordingly. And this just sort of shows for single cell sequencing, you can build that and you can identify different patterns of different cells within there. I guess just to mention that I've spoken a lot about oncology and sort of the molecular profiling, but we're also thinking about other types of data, which again could be a large expansion. So this is an example in Parkinson's disease, where we've actually started to look at digital biomarkers. So you can imagine Parkinson's disease, it's a very difficult disease in order to evaluate sort of somebody. People have huge day-to-day variability, and actually constructing sort of one-off single test within a visit to a physician is very difficult to assess somebody. So basically we've got things which are based on a smartphone, where there's a number of different tests which can go on, But you can also have this sort of monitoring during the course of the day. So you can monitor people's activity levels, not just the activity levels, but the way they're moving. You know, is it in short bursts? Can they only do so much? Or even is there something strange about the way they're moving, suggests they're limping or they haven't got good control over their limbs? A huge amount of information you can get from there in order to do that, to take that forwards. And of course, that sort of very much fits in with sort of the ideas we have around, I'm sure many of us in this room are wearing some sort of Apple Health device or Fitbit or something like that. We're all recording our individual movements at any particular time. It's always self-solved from the health point of view here. But again, if you're monitoring disease, understanding the response to treatments to disease, that's the kind of information which we can use very well. Other examples sort of come within the imaging world. So this is a sort of an interesting example from where they're doing retinal imaging. And the idea here being that we've actually got something which is a 3D image of the back of the retina. So very complex with huge amounts of voxels in there. And then they can do various different processing, identify various different features within that and put together information from there. And this is the kind of process which are actually very useful with new technologies like graphical processing units, and clusters of those can be very efficient at actually running. And actually to be able to use those and put those into machine learning in order to sort of be able to predict the future, so to identify their features in there which will actually predict those patients whose eyesight or condition is going to deteriorate over time. So again, I've got something which is very highly dimensional, but very concentrated amount of information in the patient, potentially even time series within the patient, which can be used to sort of look for the outcomes and the guide to treatment. I guess just sort of two quick words is that one is that we live in one of the things about the pharmaceutical industry, it's a very tightly regulated industry, quite correctly as well. And there's been a lot of interest about what the attitude towards AI would be, because machine learning type things, because what they really want to understand is a safety first. They're very cautious by nature, as again they should be. So things which are effectively a black box, they're going to be feeling uncomfortable with, because they're not going to quite understand if you put something in, quite what's going to come out in all situations. They've got thinking about the wider situation. But it's actually very interesting that they issued a guidance at the end of last year which spoke a little bit about these type of questions. And it was very sort of on the fence. It really wasn't saying one thing or the other, but it's going towards a cautiousness about these. But actually, within sort of the past couple of months, and if you look at these three examples here, one to do with sort of the identification of stroke patients. One is sort of the identification of patients at great risk in an ICU setting. One is to do with the actual identification of autism in infants. They've all come out within the past couple of months, so it sort of really seems to be an explosion of acceptance of these approaches in recent times. It's very interesting. The other thing I think I should say is that I probably sort of come across slightly glib in saying we've got this database over here, we've got this database over here, we've merged them all together, and that's all done. We also recognise that we're actually dealing with patient confidentiality. And patients' private information, medical history is about one of the most personal informations we can have about ourselves. So actually maintaining that patient confidentiality is absolutely right at the top of anything we're doing and any of our considerations. And we're very much aware that actually there can be a lot of leakage, especially when we get to genetic information unintentionally. And this is sort of an example of something someone did where they sort of linked together genetic information with surnames and actually identify individuals whose genome had been put online. And shouldn't have been really. We do actually have to balance that with the fact that there's a driver, both from sort of the regulatory authorities, but also more widely, and actually be able to share data and make it much more widely available. And that's something we're actually taking part in, actually actively promoting. So we have to balance these two extremes, but we do take the keeping patients' confidentiality as a primary driver in what we're doing, and take every aspect to take that. So I guess just what I want to say was a final quote, and this sort of comes from one of these market analysts, so you sort of take it with a little pinch of salt. But just to hide the bit at the top was Roche is now a data company that happens to manufacture drugs. So it just sort of recognises it's been seen in the outside world that actually the way the strategy is developing in getting information and getting data about medicines, about patients, about the prevalence of disease, the promotion of disease, is really driving things forwards. And this was just sort of a skeptic view. We're in this nice calm water at the moment, but we do see this torrent of information having to deal with that going forward. So, thank you for listening.
Thank you, Chris. That was fascinating. We have time for one quick question while we're changing slides, but there'll also be a chance, I hope, for a discussion at the end, or obviously open lunch too. Is there a quick question, or should we hold fire? Wait, no question. There we go. Sorry.
So many questions. But to pick one, how do you balance the extensiveness of real-world electronic health records with the very specific niche of mobile devices used in health? And do you think some of the reasons why the FDA are warming more towards that is that their use now is clinical decision support rather than, the final decision? So using it's one asset that the GP can use.
Yes, yeah. I mean, I think that's a good point, and I think it's the limiting thing as opposed to something which is the decision point of view. There is kind of wording which has been in the past, though, because which has sort of said that you allow the clinical decision support, but it should just be really helping what you just be doing automatically by hand anyway. So if it's something which is effectively creating additional knowledge because it's combining things together in a non-obvious manner, which sort of levels of patent which you think you'd have to get for any of these things, then it should be adding something to it. And then I think it's always difficult because it's always a case, any physician to be taking a lot of information into account, but how do you weight those individual bits? And if he believes this because it's sexy and new and comes from a nice computer and with a great name, then you would be given that a lot of weight. So
Thank you. So let's thank Chris again.